right, so let's talk about some special cases that don't exactly follow Mendel's rules. So for example, in this chicken, you see the comb, the thing on the top of his head. It's not just one size or the other. There's actually 4 different outcomes there. Or what about this, where somebody has 2 different colored eyes? So one of these cases that you might see is what's called incomplete dominance. So when Mendel did his crosses, if you remember, plants were either tall or short. That was it. And the seeds could either be green or yellow. That was it. So there were only 2 options, and you didn't ever get anything in between. Like, it wasn't like crossing tall and short, you got medium. But not every trait behaves that way. So traits that behave like Mendel's are called complete. That's called complete dominance, where all the offspring look like one of the 2 parents. But in some cases, you have F1s, the hybrids, the ones from the 2 parents, and you get a characteristic that's in between the 2 parents. And this is called incomplete dominance. Now, again, understand, if a trait shows complete dominance, like Mendel's pea plants, and the plants could only be tall or short, you would only ever get tall and short. You're not going to get anything else. But in some other kind of plant, there might be incomplete dominance, where if you cross a tall one and a short one, the offspring that are big T, little t, are medium height. So they're not tall or short, there's something in between. So this would be a very specific thing for a specific species. So for example, in this case, these flowers, when a red plant is crossed with a white flowered plant, the offspring have pink flowers. So this would be an example of incomplete dominance. And this actually happens in a species called Japanese 4 o'clocks, happens in some other flowers as well. But again, this didn't happen in Mendel's plants. So this would not happen. If this is going to happen, it would be specific to a specific type of plant that does this, where the genes show incomplete dominance and neither allele covers up the other one, so to speak. Now, since neither one is dominant, they don't always use big B and little b. Like, that doesn't really make sense. I mean, if I use big R for red, and you could, I mean, if you were trying to do a Punnett square and you wanted to use like big R for red and little r for white, and then you just had to remind yourself that big R, little r is not going to be red, because that's what would happen if it was dominant, but it was going to come out an in-between color, pink, you could do that. But typically what you're going to see in published literature is they're going to pick one letter, like in this case C for color, and they're going to use CR for red and CW for white, and then the hybrids, CR-CW, will be pink, that in-between color. So checkpoint question, why doesn't this cross support the blending hypothesis? So this is what they believed before Mendel. They said, oh, if you cross red and white, they blend together and they make pink. Well, that hypothesis would have said that then if you crossed pink with another pink, that all the offspring would just come out pink forever. That the traits blended together and pink was like a brand new gene. Well, the reason we know that's not true is because if we look at the F2, when we cross 2 pinks, notice how we get red and white back again. So that proves that blending is not what's happening. We're seeing an in-between in the heterozygous ones, but big R, when it's homozygous, is still going to be red, and little r, when it's homozygous, is still going to be white. The codes on those didn't change. It's just that the combination is giving us an in-between color. And again, notice here they did use big R and little r, even though, like I said, a lot of times that's not what they're going to do in literature. They're going to use like CR and CW. And this is another example in humans. This has to do with hypercholesterolemia, or having high cholesterol. So if you're big H, you have a lot of receptors to take in cholesterol, and you would have actually low cholesterol. You would not have a cholesterol problem, high cholesterol problem. People that are homozygous recessive, here, these people, they don't have any receptors, and they actually would have extremely high cholesterol, to the extent where they could have heart attacks even as a teenager, like it could be that bad. But if you're heterozygous, big H, little h, you have a little bit high cholesterol. You have less receptors to remove cholesterol from your blood than somebody who's homozygous, but you still have some. So you get something sort of an in-between sort of phenotype. Okay, now this is called codominance. This is a little different than incomplete dominance. A couple of examples of this. Phenotype, the ABO blood groups, which we'll talk a little more about in a second. And also this picture, this is a chicken with codominance. So codominance, how does that differ from incomplete dominance? Well, in codominance, you don't see an in-between. You actually see both at the same time. So if crossing black and white made this chicken gray, well, that would be incomplete dominance. You're getting an in-between color. But if you're seeing black and white spots, you're actually seeing the code for black and the code for white at the same time. So that's called codominance as opposed to incomplete dominance. And blood types show this too. There's an allele called IA. It stands for immunoglobulin A, and there's one called IB, and they are codominant. If you have one copy of each, IA, IB, your blood type will be AB, which basically means your blood cells will have an immunoglobulin called A, and they will also have a different immunoglobulin called B. So that is called codominance. Very similar to incomplete dominance. Again, if this was incomplete dominance, you'd get gray. Here's two more examples of codominance. This is called a roan cow on the right. When you cross a red and a white cow, you get spots of red and white. If this was incomplete dominance, the cow would be like a tannish, pinky color. And this flower on the left, when you cross purple and white, in this specific species of flower, you get purple and white speckles. You don't get a pinkish lavender flower. You get speckles of both. So codominance, you see both at the same time, like spots, speckles, stuff like that. Incomplete dominance, you see it in between. Okay. Moving on. So they want you to, here they're going to talk about multiple alleles. So this is actually the last thing we talked about today. So blood type is also an example. It was an example of codominance, but it's also an example of something called multiple alleles. So sometimes there's more than just two options. So, for example, in all the traits we've done so far, you either had big B or little b, that was it. Or big T or little t, that was it. Or CR or CW, and that was it. Well, what if there's more than just two things you could get? In blood type, there's three. So you could inherit IA, or you could inherit IA, little I. Again, the I stands for immunoglobulin, and it's immunoglobulin A. It makes a particular protein on your red blood cells that they named A. And if you're either one of these, your blood type, if they tested your blood type would be type A. If you are IB, or IB, little I, your blood type will be B. If you are IA, IB, your blood type will be AB. And if you are little I, which is recessive, you will be blood type O, which happens to be the most common blood type in this country, even though it's recessive. And now what does that mean? Not much. I mean, blood type doesn't make you more likely to survive anything. The only thing about blood type is you have to be compatible when you receive blood from somebody. If your blood type is, let's say, A, and you receive blood from someone whose blood type is B, it'll cause a really bad reaction, like it could probably kill you. So if you ever receive blood, you have to receive from somebody who's a match. But since O is recessive, people with type O, they don't actually have any of those proteins on their blood cells, and so they won't cause a reaction in anybody. So they're sometimes called the universal donor. And we're not really going to focus on that, but today we're more talking about the genetics of it. But O is the universal donor. They can donate to anybody because they don't have this protein A or B on either of their blood cells. Okay, so checkpoint question. Maria has blood type O. That means she has to be little I, because that's the only code that would make her O. Her sister is AB, so her sister is IA, IB. So the question is, what is the genotypes of their parents? Well, since Maria is O, we know that both of her parents, mom and dad, both must be carrying a copy of little I, right? That's the only way Maria could be type O. And that means that the other letter for each of the parents, one must have A and one must have B. And maternal grandparents means the grandmother on the mom's side. So they're saying that basically her grandmother, her mom's mom, had blood type A. So the mom must be the one who's IA little I, and the dad would be IB little I. Now you might say, well, why couldn't it be that one of the parents is this and the other one is this? Well, because remember, you don't inherit both your genes from one parent. If Maria is blood type O, little I, she had to get one of these from this parent, and the other one had to come from the other parent. So both parents are involved in this. So an AB parent cannot have a type O child because they can only give either A or B. So that's why that would work that way. And I believe this next slide here, whoopsie, sorry. So this is just kind of showing you what would happen. So when they show the blood clumping together, that would be a bad reaction. That would mean that that you could actually die. So if your blood type was A, but somebody gave you blood type B or blood type AB as like a blood donation, a transfusion, because you were in an accident and needed blood, your blood would all clump together like this inside your blood vessels, and it would probably kill you. So a person who's A can only receive from A or O. A person who is B, they can receive only from O again or B. A person who's AB, look at that, they're lucky. They can receive from anybody. And then a person who's O, unfortunately, can only receive from O. So that just gives you kind of an idea. You're not going to be tested on who can donate to who. We're really talking right now about the genetics of it, but a lot of people are curious about that kind of thing. All right, and that's where we're going to stop for today.